Can we started? Hello everyone. I hope that you all are having a great time. You once again are ready to learn something new. And today we are going to talk about a very important chapter of class 11's biology, photosynthesis. So we are going to start with the basic introduction of the chapter and we'll talk about a few experiments, right? But before that, let us quickly talk about this new segment that the Unacademy has launched to help you ace your term 2 examination. Now Unacademy has launched classes for ISC, I always mess it up, ISC and ICSC, ICSC boards, right? So we have classes for 9th, 10th. Hello, Devil OP. Oh my god, what a name. How are you, bro? Are you ready to study some photosynthesis? Right, so we are going to talk about photosynthesis, arguably the most important topic not only for the syllabus but also for the whole knowledge of biology itself, right? It is the process which gives energy to the biosphere, right? I'm replying to you. Hello. I can't reply in the chat. I have to reply in the video. So yes, tell me, how are you and are you excited? Or basically you must have some idea of the process of photosynthesis. Today we are going to just develop on those ideas. We are going to recall all the things we already know and it will give us a basic foundation for all the things that we have to study. So I'm going to bifurcate the things and I'm going to make the chapter very easy for you to understand. And there are other excellent teachers which are in this new segment which will help you ace your ISC board examination for all these classes. And if you want to get extra advantages, fine sir, actually I'm not bio student, still I have subscribed. This is called love. Nice. Okay, but bio is not a very hard subject. If you laughing emoji is fine, just keep the vibe cool, right? So yes, I was talking about the plus subscription. So you can really ace the examination if you have the plus subscription with you. Everybody knows it. There are great features of the plus app. The most important feature of the plus are the courses. And me myself and my other teachers, my partners, we have all launched a crash course to help you all ace the examination and get great marks. I can't talk. Get great marks in your examination and revise the whole term 2 syllabus. So we have introduced the crash course and you can join the crash course if you have the plus subscription. And you can now buy plus subscription with a 10% discount. All you have to do is use a magic code, and that code is this SOURAV, which is my name. Let me introduce myself. My name is Sourav Verma and I will be your biology educator. And the code that you can use is Sourav B Live. So this is the code that you can use to get 10% discount whenever you avail the plus subscription. Okay, let's move on. This is me, right? And I'm Sourav Verma, your biology educator, and at the moment I'm pursuing MBBS. So I will be teaching biology to class 11th and 12th. Alright, let's move on and let's talk about today's topic. So the topic of today's class is going to be photosynthesis. So we are going to talk about the very basics of photosynthesis and the discoveries that were made during the understanding of photosynthesis by all the different scientists, right? So without further ado, let's get started with the discussion, right? So yes, you can follow the Unacademy Twitter, Telegram group as well. So the link is going to be in the description. Check it out, and the Telegram group will provide you with all the materials and all the updates related to all the classes. And yes, if you're liking the videos, then you can subscribe the channel. We are targeting for 1,000 subscribers. let's let's set it right. So now let's talk about photosynthesis. Let us get an overview of this wonderful process, of this wonderful phenomenon which is capturing the energy from sunlight. So let us get basic idea of photosynthesis. So the green plants, they carry out photosynthesis. So the word photosynthesis itself means, photo means light, right? Photo means light and synthesis means formation. Formation. So the process of formation of food or organic compounds using the energy of light is known as photosynthesis. It is a physico-chemical process. What is this term actually meaning here? See, physico means related to physics or physical. Physico means physical. The study of physical phenomenon is physics, and chemical. So this is a physico-chemical process. Every biological process, in fact, is a physico-chemical process, because if we zoom in, then we'll observe that there are the things which are happening, the changes which are happening on the microscopic level in biology are actually physical changes and chemical changes. Certain chemical reactions are happening, certain physical changes are happening, and the whole mixture of all these changes, they lead to a biological process or a biological change. So photosynthesis is a physico-chemical process in which we use light energy from the sun to drive the synthesis of organic compounds, right? Organic compounds are the compounds which are made up of carbon basically, right? So light energy is used to make organic compounds. So ultimately all life forms on the earth, they depend on sunlight for energy. See, all the energy coming from the sun is used by us. The energy from the sun comes in two forms, right? So the sun, it gives us energy in two basic forms. First is the light energy and second is the heat energy. And this light energy can be captured by green plants to carry out the process of photosynthesis. Okay, and it says that the use of energy from sunlight by plants doing photosynthesis is the basis of life on earth. So yes, it is the procedure, it is the phenomenon that is enabling life on the planet, because energy is very important to carry out all the process. And all the materials, all the organic materials that you see, they are also formed by the process of photosynthesis. All the fossil fuels, and yes, all the oxygen as well. The oxygen, which is very important for our survival, without which we cannot survive, is also a result of photosynthesis. So let us understand what are the significance of photosynthesis. So photosynthesis is important due to two reasons. First, that it is the primary source of food on earth. Everybody knows it, that photosynthesis makes glucose, and using glucose all the other biomolecules are made, and those biomolecules, they make up our food. So it is the primary source of food on all earth. Other than photosynthesis, there is only one other process which can actually capture energy or create food, which is known as chemosynthesis, right? So I should write it down. Chemosynthesis, it is the only other process which can capture energy in the form of food, and this is performed by some bacteria. So some bacteria which live in extreme conditions where sunlight is not visible, it is not present, so they can use chemical energies to make their own food, right? So this is chemosynthesis. This is the only other process which can produce food and using a chemical energy. All right, and the second significance is that it is responsible for the release of oxygen. So photosynthesis not only prepares food, but it also releases oxygen using carbon dioxide, right, into the atmosphere, because oxygen is necessary for respiration in all the animals. So let us understand photosynthesis using its equation, because it is a chemical change, so it must have an equation, and we have an equation. Now I will write two equations. Let's see. Let me first write the reactants. So we have carbon dioxide, which is a raw material of photosynthesis, and we have water, another raw material. These two combine and they form glucose, which is C6H12O6, and with that it forms oxygen, right? So this is the unbalanced equation. Now let us balance it. So to make one glucose molecule which has 6 carbon, we must use 6 CO2 molecules, right? So this is balanced. And if we use here 6 water molecules, we will have how many oxygens? I guess 6 oxygens, right? So this is the balanced equation of photosynthesis. Now it is correct chemically, right? So it is the chemically correct equation, or we can say correct for chemists. This is correct for chemists, those who study chemistry. Why? Because it is properly balanced, and this is what is happening. We are using CO2 and water, and what we are getting? We are getting glucose and oxygen. We are also getting some water here, but see, some water was produced on this side, but it got cancelled, right? So this is the balanced equation for the chemists, but for biologists it is not very correct. Something has to be changed to make it biologically correct. Let me write the same equation again. So we have CO2 and H2O, and they make C6H12O6, and they make oxygen, but they are also making water. They are using water as well as they are making water. Both of these are different, and the equation is balanced in a different way for biologists. Let me show you. So yes, 6 CO2 are used to make the glucose, but now the water which is used actually has 12 molecules, right? So 12 molecules of water are used, and I will tell you why it is significant. And 6 water molecules are released here, and 6 oxygen molecules are released here. So you see, any chemist would cancel 6 water with 6 water here, and we'll get to this equation. So this is the chemically correct one, but for a biologist the correct one is this. So this one is correct for a biologist, because this equation is actually expressing the whole series of reaction, because this equation is not representing a single step reaction. It is representing the whole pathway which is forming glucose and oxygen. Now I will tell you why this and this are different, but let us first know that the energy source is going to be light here, right? So the energy source is light in this as well as in this, and because we are taking in energy to carry the process forward, this particular reaction has to be endothermic reaction. Can you remember endothermic, which means the reaction which moves forward by taking in energy? We represent it as this, the delta H. The change in enthalpy in this case is positive. It is something related to thermodynamics. Because we are taking in energy to carry the process, this whole process is endothermic. It takes in energy in the form of light, so it is intaking light energy. Another term which is used to express endothermic is endergonic. So yes, again, this is the term used for endothermic. So these two terms are used to represent endothermic or endergonic reaction, and photosynthesis is endothermic. Why? Because it is using light energy to move forward. Now let us understand why we need to write water here as well as here. See, all these 6 carbon atoms, they go into the glucose molecule. So I am tracing the path of all the elements to know where they are actually going. So in the reactants we have CO2, and the products we have glucose. So these 6 molecules of carbon, they go into these six atoms of carbon, they go into the 6 atoms of glucose. Now we have 12 oxygen, right? O2 times 6 is 12 oxygen. Now where are these 12 oxygen molecules going? Arguably the 6 molecules are going here, but the remaining 6, they can be here or they can be here. And this was a confusion for all the scientists. They wanted to know whether or not oxygen in the carbon dioxide is the source of oxygen released in water, or is oxygen released in photosynthesis, or the oxygen of water is released here. And it was found out that this oxygen is actually this one, right? So let me color code it, that this particular oxygen on this side, it is not making the oxygen molecule released in photosynthesis. This oxygen is actually released in the water molecules, right? This is the first thing. Second thing, the oxygen which is actually released during photosynthesis, these 12 atoms of oxygen, all of them are coming from water, right? So as you can see, this is how it is happening. And this hydrogen, it is going in glucose as well as in the water so produced, right? So this is how the atoms are moving. And let us get an idea of the significance of all these reactants and the products of photosynthesis. See, CO2 here, it is acting as carbon source. So CO2 is the C source, carbon source of the reaction. Water, what is the role of water in photosynthesis? It acts as electron and proton donor. Electron is E minus and H plus, which is hydrogen, nucleus is proton. So electron and proton donor, which means it gives electron and protons to carry the reaction forward. What is the role of electron and proton? It will be studied later in the chapter. So these two are used. Now, this is the main organic product formed. This is glucose, and this is the product, organic product of photosynthesis. Organic product of photosynthesis. And oxygen is the gaseous product of photosynthesis. So yes, oxygen is the gaseous product and some water is also released. So this is how we can understand photosynthesis in a better way, and this is the biologically correct equation to represent photosynthesis. Alright, now let us see where all these things are coming from. So CO2 comes from environment, right? It comes from atmosphere. It comes from atmosphere. Water, it comes from soil. That is why we need to water the plants. So it comes from soil. This light is captured with the help of certain pigments, right? So we have to use certain pigments which capture the light energy. And this glucose, it is produced in the leaves, but it is stored in the form of starch, right? So this is the organic product which is stored in the form of starch. In the form of starch. Starch is a type of carbohydrate. Although glucose is a simple carbohydrate, starch is actually a polymer of glucose. I should add it here. It is a polymer of glucose. Polymer of glucose. So why do we need to change glucose into starch? Because glucose is chemically reactive. It can react and change to some other form. But starch is chemically non-reactive. So converting glucose into starch is actually beneficial because it remains put and it does not change anything into anything else. And that is why we always check for starch. We perform iodine test to check for starch whenever we want to see whether photosynthesis is happening or not, right? And oxygen is the gaseous product which is released in the environment through stomata. So this is the basic idea of the chemistry of photosynthesis. It is not done though. let me give you some features of photosynthesis that will make the picture better. So some features of photosynthesis is what we are going to talk about. So the very first feature is it is endergonic process. Endergonic process, which means that they take in energy, right, in the form of light. It is a endergonic process, which means use energy, use light energy. They use light energy. All right. The second feature of photosynthesis is that they are anabolic process. Anabolic process. I'm sure that you know what anabolic and catabolic processes are. Anabolic processes are synthesizing process which uses simpler substances, joins them and make bigger substances. So yeah, anabolic process means synthetic process, which means they synthesize something. Synthesizing process, let's write it like this: synthesizing process. What does it synthesize? Glucose. Synthesizing process. So it is anabolic process. The third feature of photosynthesis is that it is a redox reaction. Now, redox reaction are something that you must have studied in chemistry. It means oxidation and reduction. The basic idea is that oxygen is addition of oxygen or removal of electron or removal of hydrogen atom. I'm sure that you know it. Let me give you an idea that redox is made up of two reactions, oxidation and reduction. So oxidation, it can mean addition of oxygen, or it can mean removal of hydrogen, or it can mean removal of electron. So all these, they represent oxidation. On the other hand, we have reduction. And reduction means either removal of oxygen, addition of hydrogen, or addition of electron, right? So redox reaction are the reactions which are occurring in the photosynthesis. Alright, so let us study how it actually happens. What substance is getting oxidized and what is getting reduced. So you see, CO2, it forms C6H12O6, right? In this case, carbon is getting reduced, right? So carbon is getting reduced. This is reduction. And on the other hand, we have water, which is converting into oxygen. And because hydrogen is getting removed from oxygen, the oxygen is getting oxidized, right? So these are the redox reactions occurring in the photosynthesis. That is why it is a redox reaction. So these are the properties of photosynthesis. Okay, now let's move on. And now let's talk about a few experiments that gave us a better understanding of photosynthesis, right? So the first experiment is the experiment which express the importance of sunlight and chlorophyll pigment. Yes, the green colored pigment which makes all the leaves appear green is the chlorophyll pigment. So let us talk about it. So this first experiment, it will tell us about the significance of light and the pigment, basically chlorophyll. Let's see. So the experiment to look for starch formation, again we are looking for starch formation in this process in two leaves. So we have taken two different leaves. First is a variegated leaf. What does a leaf means? A leaf which has these patches, uncolored patches, which are not green, right? So variegated leaf is a leaf having non-green patches, non-green patches. Of course, these patches, they do not contain the chlorophyll pigment. That is why they are not green. So first we have taken variegated leaf and we have taken a simple leaf and taken that leaf and covered it with a black paper. So either you can take a variegated leaf or you can take a simple leaf and cover it with the black paper and then expose it to light. On testing the leaves, we first take a simple leaf and then we take a variegated leaf or a covered leaf. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of sunlight. So it was clear that photosynthesis only occurs in the green parts of the plant and it only occurs in the presence of sunlight. So this is the first experiment that gave us an idea of the significance of light and the pigments for photosynthesis, right? And the second experiment, which is also known as the moles experiment. Moles experiment, it gave us an idea of the significance of CO2, right? Carbon dioxide. So let us see what this moles process is. So a part of the leaf is enclosed in a test tube or in a beaker, as you can see, and that test tube contains KOH soaked cotton balls. Here we have the liquid KOH, or we can have cotton balls which are soaked in KOH. Basically we need KOH. Why? Because it absorbs the CO2 of the environment. So all this air inside this beaker or this test tube, it does not contain CO2 gas, while the other half is exposed to air, as you can see. Now the setup is then placed in light. So we place the setup in light for some time. On testing for the presence of starch, again with the iodine test. So we use I2 test to test for starch. If the starch is present, this gives bluish black color, right? Bluish black color. It would mean, what would it mean? That starch is present. Starch present. And if no color is seen, no color change is seen, which means starch is absent, and starch is formed due to photosynthesis. See, photosynthesis forms glucose and that glucose is converted into starch, right? So on testing for the presence of starch, later in the two parts of the leaves, the exposed part of the leaf, it tested positive. So this is the positive test when it turns bluish black for starch, while the portion which was in the tube, it tested negative, which means without CO2 photosynthesis is not possible, right? So this showed that CO2 is important for photosynthesis, as we have already seen that CO2 is the main reactant or the raw material used for photosynthesis. This is the one which makes glucose and that glucose forms starch. So yes, this experiment is proving the significance of carbon dioxide for the process of photosynthesis. Alright, now let us quickly talk about all the experiments and all the results of those experiments related to photosynthesis given by different scientists. So the first scientist is Joseph Priestley. You should know the name of Joseph Priestley because he was the person who discovered oxygen, and he also discovered something important related to photosynthesis. Let us talk about it. So Joseph Priestley in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. So it said that air is important for the growth of plants. Priestley, you may recall, he discovered oxygen. As I told you, he discovered oxygen in 1774. So, what were the experiments that he conducted? So, Priestley observed that a candle burning in a closed jar, like this is the closed jar, and if we have candle burning inside it, it soon gets extinguished, right? So, if you only have the candle in the jar, it will soon extinguish. Similarly, if we have a mouse, it would soon suffocate in the closed place and it can die, right? So all these things, they are undergoing changes when they are placed in a closed jar. The candle extinguishes and the rat or the mouse, it dies, right? So how is it happening? He said, based on his studies, he concluded that burning candle or animal which is breathing, that breathe the air, they somehow damage the air. So this was the basic thought that came to his mind based on the observation that these organisms or the candle, they are polluting the air, they are damaging the air somehow. And when he placed a plant, which was the mint plant which he used for his experiment, he placed the plant and he placed the candle and the mouse, right? He found that mouse stayed alive and the candle continued to burn. So plant was basically doing something which was keeping the candle burnt and the mouse alive. So it was something magnificent. Plant is doing something. So Priestley hypothesized, he gave an explanation to all these experimental observations. He said that plants restore to the air whatever breathing animals and the candles remove. So his idea was that the air contains something which keeps us alive. That is why we need to inhale air, and that is why candle also needs air to burn, right? So his idea was air contains some things which keeps us alive. When we breathe the air, we damage that particular thing, right? And similarly, candle when it burns, it destroys that particular thing which was keeping it burning. But when we place the plant, that plant is actually replenishing, it is creating that particular thing which keeps us alive, and it basically restores the quality of air to keep us alive. So yes, these were the experimental observations by Joseph Priestley, and he basically told the release of oxygen from photosynthesis, that oxygen gas is released from the photosynthesis process, and this oxygen is important for the burning of candle as well as for our respiration. So this was Joseph Priestley. Another scientist was Jan Injun House. Now Jan Injun House used similar experiments related to Joseph, which were used by Joseph Priestley, but he found out the significance of sunlight for photosynthesis. See, using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight, Jan Injun House showed that sunlight is essential to the plant process, for the survival of plant, for photosynthesis, and that particular process which purifies the air fouled by burning candles and breathing animals. So he said that the whole procedure of restoring the air which is performed by the plants, it is only possible in the presence of sunlight. Sunlight is important for whatever the process which is happening in the plant, right? So his experiments explained the significance of sunlight. Significance of sunlight. Right? Also, Injun House, in an elegant experiment with an aquatic plant, showed that in the bright sunlight, small bubbles were formed. So, if the aquatic plant, it is kept in bright sunlight, small bubbles appear on the green parts, around the green parts, while in the dark, no bubbles appear. What were those bubbles of? Those were the bubbles of oxygen. So you can imagine in the beaker, if we have water, in the water if we have any green plant placed, in the green parts of the plant, if sunlight is provided, if sunlight is provided, what did he observe? He observed tiny bubbles, right? Tiny bubbles were formed, right? And these bubbles are of oxygen. So these are the bubbles of oxygen gas, right? But in the absence of sun, in dark, no oxygen bubbles were formed. So he gave the significance of sunlight for the oxygen process, or this whole photosynthesis process. Okay, let us move on. Now let's talk about the third scientist, which is the Julius Monsex. He told us that glucose is formed during photosynthesis. Is formed during photosynthesis. So this is what he told us. How did he do that? Let's see. So in 1854, Julius Monsex provided evidence for the production of glucose when the plants grow. So when the plants are growing, they make glucose. His later studies showed that green substance, some green substance in plants, which we today know as the chlorophyll. As we know it now, it is located in special bodies. What are those special bodies? Today we call them chloroplasts within the plants. So he just gave an idea that glucose, it is formed in these with the help of these green substances which are present in certain structures found in the leaves, found in the plant cells, and those structures were chloroplast. He did not give any term like chloroplast or chlorophyll, but he had an idea that something like that is happening. Today we know that green pigment is the chlorophyll and that structure is the chloroplast, which is an organelle found in the plant cells, and these organelles perform the procedures of photosynthesis. Alright, so he found that green parts of the plant is where the glucose is made. So he found that out, and that glucose is usually stored in starch. As I've already told you, that glucose is made but it is stored in the starch. Why? Because glucose is reactive, glucose is soluble in water, but starch is not reactive and not soluble in water. That is why it remains where it is stored. That is why starch is more suitable for storage. To give you an idea of all the forms, see, glucose is the sugar which is formed. Glucose is the one which is formed. It is transported in the form of fructose. No, sorry, sucrose. Sucrose. So if we have to transport sugar, we transport it in the form of sucrose. Right? Sucrose is suitable for transport. Sucrose is a disaccharide which is made up of glucose and fructose. Right? So sucrose is made up of glucose plus fructose. Right? And for storage, it is stored in the form of starch. So starch is the form used for storage. So this is how all the different types of sugars, they are used by the plant. I hope this is clear to you, and I hope the studies of Julius Monsex are clear to you. Let us move on. Now we will talk about T.W. Engelman. So he conducted some important, interesting experiments. So what he did was, he split the sunlight using a prism. We know that prism can split white light into its constituents color. It can make the wave cure, right? So he split the sunlight using the prism, and he used this cladophora. Cladophora was in water algae and some aerobic bacterium to find out which particular part of light, which particular color of the light is used the most by the plants. That is what he wanted to know. So his experiments were very interesting. Let us see. So using a prism, he split light into its spectral components. So he split light in all its colors and then illuminated green algae, which was cladophora. Again, you have to remember which particular algae was used. So he illuminated the green alga, which was placed in a suspension, a water solution of aerobic bacteria. As you can see, in a beaker, this cladophora was used and some aerobic bacteria were used. Aerobic bacteria are the bacteria that require oxygen for survival. So you can imagine, wherever there is production of oxygen, these bacteria will appear more in number, right? So he wanted to know at which particular color, at which particular wavelength of light is the plant most active, where the plant is forming the maximum amount of oxygen. Let's see. So the bacteria were used to detect the sites of O2 evolution. So where there is more bacteria, there will be production of more oxygen gas. So he observed that bacteria accumulated mainly in the region of blue and red light in the split spectrum. This is the main observation of the whole experiment, that most of the bacteria, they were found in the bluish part and in the reddish part of the whole spectrum, which means most oxygen is produced in these regions and plant is most active at these particular wavelengths. As you can see, this is the wavelength close to 700 nanometer, and these are the wavelengths close to 4 or 450 nanometer, right? And 700 nanometer is the wavelength of red color, and 450 nanometer is the wavelength of blue color, right? And this is the action spectrum, which was given by T.W. Engelman. So a first action spectrum of photosynthesis was thus described, right? So these were the experiments performed by Engelman, and this is the action spectrum which was observed, that most of the oxygen or most of the activity of the plant is seen at blue and red light. So it is very significant. Alright, let's move on. And now we will talk about the experiments of Cornelius Vanneel. Cornelius Vanneel told us that oxygen, O2, comes from water, from H2 and not CO2. Earlier it was thought that CO2 gives oxygen, but this oxygen comes from water. So how did he find that out? Let's see. So a microbiologist was Cornelius Vanneel. So Cornelius Vanneel, based on his studies of purple and green bacteria, he demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. So it is basically saying that CO2 will convert to carbohydrates, C6H12O6, let's say glucose. Now it is a form of reduction, and for this reduction plants use a suitable hydrogen donor, right? So this hydrogen, it reduces this particular CO2 into glucose, and this hydrogen can come from any suitable donor, right? In bacteria, this hydrogen donor, in the bacteria which he was studying, the hydrogen donor was H2S, hydrogen sulfide, right? And in plants we know it is H2O. So in bacteria which he was observing, the hydrogen donor was not H2O but it was H2S, right? Okay, so let me tell you, it was H2S. And if you see the whole equation of the reaction in bacteria, in bacteria in bacteria the equation comes out to be like this: CO2 with H2S gives C6H12O6 along with sulfur, right? So 6H2S, 6CO2 and 12 sulfur atoms. So this is how photosynthesis in a bacteria works, and no oxygen is released, right? So no oxygen is released, which means that oxygen cannot come from, it cannot come from CO2 because we have given CO2 but still no oxygen is found. What we have changed is we have given H2S instead of H2O. So it proves that oxygen comes from water and not from carbon dioxide. So this was the main observation of experiments performed by Cornelius Vanneel. Alright, so yes, this is all about today's session. We have discussed all the experiments and the basic idea of photosynthesis is clear to us. Let me give you a little more details about photosynthesis. So in photosynthesis we have green parts, and the main green part is the leaf. So inside the leaf, let me tell you, so this is a leaf, and if we zoom in on a leaf we will find the cells, right? The plant cells, and the leaf is made up of mesophyll cells mainly. These mesophyll cells, they are mostly long, so let me draw long cells, right? So we have these mesophyll cells, and inside the mesophyll cells we contain, the plants contain certain organelles. These organelles, these are the chloroplasts, right? The organelles in which photosynthesis occurs. And these chloroplasts, they look something like this. So this is the whole chloroplast, which is a double membrane bound organelle found in the green cells or the cells of the green parts of the plant. And this the chloroplast, it contains such structures. I'm sure that you know about all these structures. All these stacks are known as grana, and they are joined together by these stroma lamella, right? And this is how they exist, right? And it also contains its own DNA and ribosomes and all. It is a semi-autonomous organelle. All right, and we know that this region, this whole space is stroma. This is a granum, and multiple granum are grana. So this is a granum, and a granum is made up of multiple thylakoid lamellae. Thylakoid lamellae. And these tubular structure which are joining them are stromal lamellae. Stromal lamellae. Right? Now, the thing that we have to focus on is this thylakoid lamellae. So let us zoom in on this thylakoid lamellae. So the membrane which makes the thylakoid lamellae, it looks something like this. So we have this thylakoid, and it is making the other part like this, right? and this appears something like this. And yes, all the other thylakoid, they appear, they are arranged something like this, right? So this is how they are arranged one over other, and this arrangement gives us two different regions. First region is this, which is breast, this region, all these regions, they are oppressed, so they are called oppressed region, right? They are not open, so they are oppressed region. And this region is the open region, right? They are non-oppressed region, something we will talk about, right? Now if we further zoom in on this part of the membrane, then we will observe that this, let's just say this is the whole membrane zoomed in, and we observe certain complexes, complex here, complex here, a complex here, and complex here, and another one here, right? So certain complexes, they are arranged, and these two bigger complexes, these two systems, they are responsible for capturing of sunlight. And this particular complex, it is responsible for taking water and producing oxygen, right? If we take one water molecule, we'll get one oxygen, right? So how this whole procedure is occurring, this is something which we will discuss in the light reaction. Again, photosynthesis is distributed into two parts. One is a light reaction, another is the dark reaction. So light reaction occurs in the membrane of thylakoid, and dark reaction occurs in the stroma. So in stroma we'll talk about the dark reaction, right? So this is how photosynthesis works, right? So I have given you enough idea related to photosynthesis, and further details about it will be discussed in the next class. So this is it, and let me tell you some great features of the subscription. If you take the plus subscription, what are the features that you will get? First of all, you can learn live, right, from the comfort of your time. अपने घर पे आप पढ़ सकते हो. There is no issue, you don't have to go anywhere, and also the time is not good to go out. Omicron variant is on the rise and next wave is coming, so all these restrictions, they won't allow you to go anywhere outside. So yes, you can use this Unacademy platform to learn all the things very well. Second, unlimited access to all the courses. Once you get the plus subscription, you can watch any plus class, any course which is started by teachers. So all the classes, they will be visible to you. Top educators of India are all on the same platform. So all the top educators will be teaching you all the subjects, and you can choose between different educators as well. Regular doubt clearing and answer writing sessions are also conducted, so you do not face any problem, any confusion related to any concept. Exhausting coverage of syllabus. So the whole syllabus is completely covered multiple times, so you don't forget even the slightest details related to the topics, right? Mentorship and guidance as well. So it is not only about the subject, there are thousand other things which a student goes through, and we understand it. You can talk about us, we can talk with your parents as well. So any problem that you're facing will be solved by the educators of Unacademy, right? You will get all the material. For example, all the notes that we show you in the classes, they will be all yours. You can download them and you can revise with them anytime you want. You will also get practice tests, so you can challenge yourself. You can basically test your knowledge. You can test whether or not you have understood everything, and if there is a problem, the teachers are always there to help you. We have live test series as well, So it creates a lot of excitement when there is a test series and different students, they are competing for the great scores, and it will help you, it will motivate you, it will push you to work harder. And we have batch courses as well. So all these great features can be available to you if you take the plus classes. And if you're thinking about taking the plus classes, you can use this code and you will get a discount of 10%. All right. Yes, when you're taking the course, these are all the courses that you can take. We have 6 month, 9 month, 12 month, and all these courses. And something that you will observe, that if you take a long-term course, you will have to pay less amount per month. So yes, if you're thinking about taking a new plus subscription, and if you want to take a long-term course, that will be a lot beneficial, because you will save a lot of money. It is very advantageous. And if you want to save 10%, yes, you can use this code. All right. So let me give an idea of all the courses which we are introducing. So we are introducing the ICSC Class 9 Hindi course, where you can study in Hindi, and all these great teachers will be teaching you all these subjects. We have maths, physics, chemistry, and biology. We also have a course in English language, which is known as the champion course for all the subjects like maths, physics, chemistry, and biology. We have for class 10th as well in Hindi as well as in English, and also for class 11th we have maths, chemistry, and biology. I'll be teaching you biology there. And we also have a champions batch for class 12th as well. So yes, 9th, 10th, 11th, and 12th, all these classes are covered, right? So these are all the teachers and all the subjects and all the courses that you will be having if you have the plus subscription, right? And also you can use the free classes at Unacademy to see how you are settling in with the environment of the online teaching, and you can see basically which course you have to take for the plus classes. So it is a nice demo of all the things that you can get with the plus classes, right? So how you can watch the special classes? First you have to download the Unacademy app from the Play Store. Get started, fill your mobile number and email ID to get all the notifications. After that, select the class, select the board, right, or any competitive examination, select the language, and your whole experience will be customized according to that, right? And we have this in the description, we have links to the special classes which are coming in, and you can go to the app and you can click on notify me. So whenever there is a live class, you will get notified, so you don't miss any class. And you can use the code to get the special classes for free. So the code is again SOURAV be live. So use this code to watch the special classes for free, right? So yes, for the recap, we just talked about the introduction of photosynthesis. So nothing heavy was done here, we just understood photosynthesis. So we talked about introduction of photosynthesis. All right, so I hope you enjoyed today's class, and I will see you in the next one. Download the Unacademy app, get started, and crack it. All right, bye bye, and I will see you